Guys, welcome back to my channel, and today we'll be discussing about the bacterial growth. As you know that in higher organisms, growth refers to an increase in size and volume. But in case of the bacterial growth, it refers to the increase in number. It takes place by binary fission, that is a form of asexual reproduction, where a single cell organism divides into two cells of the same size. And the bacterial population grows in a geometric manner or the exponential manner, with each generation producing 2 cells, 4 cells, 8 cells, like in, you can say like this. That is, in the first phase it is 2 cells, in the second phase it is 4 cells, and it is 8 cells, and so on. So uh, likewise, the bacteria needs certain conditions to grow, that are the favorable conditions that are not same for all the bacteria. So factors may be oxygen, pH, temperature, light affect the growth of organisms. Likewise, osmotic pressure, atmospheric pressure, the available moisture are also some additional factors that affect the growth of bacteria. And the growth rate is influenced by the composition of the growth medium in which they are growing. When the nutritive culture medium is inoculated with a given number of bacteria incubated for a sufficient period of time, it gives us a characteristic growth pattern to understand. The graphical representation of the number of the living bacterial cells in a population over a period of time is called the bacterial growth curve. So we'll be discussing about the growth curve, and it is divided into 4 phases. So you can see here. So what are the 4 phases? The first phase is the lag phase. That is the starting phase. So during this phase, the cells are metabolically active, but they are in the stage of not dividing. They do not divide in this stage, just they are metabolically active, and the cells prepare themselves for the division by synthesizing enzymes and various other molecules. And the second phase is the log phase, which is also known as the exponential phase, where the bacterial cells divides continuously, and these cells are the most active in this phase, and they are growing, dividing, and increasing their number exponentially. So this phase is also known as the exponential phase. And next is the stationary phase, where the growth of bacteria reaches a state during which there is no net increase in the bacterial population, as the number of dividing cells is equal to the number of dying cells. Thus, a stationary condition is prepared, and this is due to the depletion of the nutrition and the accumulation of the toxic waste products as the byproduct in the log phase. And after the stationary phase, there comes the decline phase or the death phase. That is, all the nutrition in the growth medium becomes zero, or we can say it decreases, and the accumulation of toxic waste product increases. That makes the number of dying cells exceed the number of newly born cells, so it is totally declining. This is all about the bacterial growth and the bacteria growth and bacterial growth curve. Thank you for watching. If you want me to describe any other topic, you can just comment down below. You can subscribe my channel, and have a good day. Good night.